Bet you didn't expect to see this again. We're gonna finish it. We have to. I promised you I would, so therefore we will do it. I am nothing if I am not an ape of my word. If you will recall with me, this is a text titled Why Human Evolution is False, the Scientific Case for Independent Origins. Has ape-to-man evolution been overturned? By YouTube Creationist Standing for Truth. It's got, the big font. We all remember that, poking fun at kind of the formatting of this text. And if you follow this channel, you'll know that my standing with Standing for Truth is on hold for the time being until the channel decides it prudent to answer my questions in a non-half-assed way. I listed some questions that I felt young Earth creationists need to answer for, and given I answer all of the questions given to me, I expect that to be a two-way street if someone is claiming that this is a dialogue and not like a preaching moment. So I'm not going to be interacting directly with the channel, but this series was started before that, so we will be finishing reviewing this book. We're on chapter 3 today, out of 5, which is great. We still have a ways to go, but we will be thorough. That I promised a thorough review. We're doing a thorough review. We're going to look at this text in its entirety. And to prove that I am indeed a person of my word, I got an offer to debate David McQueen over on the Standing for Truth channel. David McQueen being that one guy who thinks there's human blood in oil because of the Noachian deluge. So I passed up that opportunity for you people because you keep calling me out saying that, oh, Erica doesn't mean it when she says she's done with Standing for Truth until they answer the questions. I turned it down, so, heap praise upon me. What else before we begin? What else? This is pre-recorded. I always pre-record these so that I can do edits where needed because I tend to get kind of rambly with these books. If you like the content on the channel, like, comment, and subscribe, and, you can support the channel through Patreon or PayPal or the stores if you so desire. But of course, I plan on providing this content regardless because it's a hobby and maybe I'm a masochist. I don't know. We will continue. Yes, I remember. I was going to say one more thing. Standing for Truth did attempt to answer the heat problem, and yes, I am using the book review as a platform to complain about Standing for Truth because I don't get any other opportunities because I said I wasn't going to talk about the channel, and I'm kind of an ape of my word on that. He made this video recently. It was a debate with David Neff, who you should sub to, which was about the heat problem. It was the last evening, I believe, on his channel. It was late at night, so of course I didn't get the opportunity to hop in. Not that I would, because I told you guys I wouldn't, but it would have been tempting. And in case you're wondering, no, it did not actually address any of the real issues with the heat problem. The heat problem being, of course, that for the Noachian deluge to have occurred, it would have accrued so much heat so as to vaporize the granitic crust of the Earth half a dozen times over. If we're going on the conservative end, and on the extreme end, we would see the absolute annihilation of the planet due to approximately 1,000 trillion 1 megaton hydrogen bombs per square meter of water for the entirety of the flood. So we've got some heat problems going on. Standing repeated the same things that he repeated before my hydroplate video, which we're coming up on a year since that release. And I'm not sure what he's actually done to alter his model, because he was essentially like, space is a heat sink. This can relieve some of the heat. And then I was like, here's why space doesn't work as a heat sink. Or, oh yes, well, the water is going to be able to jet up into space by these big massive fountains. And I say, well, here's why it can't do that. You have to have a de Laval nozzle and all of these kinds of things to get that escape velocity. But of course, he's not addressing any of those critiques to the mitigations, right? Which is part of the problem. So, of course, those questions remain unanswered, and unfortunately, I cannot engage with the channel until those questions are at least attempted. I just want to see an attempt to answer them instead of ignoring those critiques. So with that, we can now begin our journey, our spiritual journey, if you will, through chapter 3 of Why Human Evolution is False by Stating the Truth. Now, chapter 3 is called Universal or Separate Ancestry. We have a nice picture of the patriarchs there. I really like this graphic here. Of course, I don't think Standing made that himself. There's a lot of cribbing going on, a lot of kind of snatching graphics and things of that nature and putting them into the book, along with those hyperlinks that are, of course, unclickable to me, given this is a paper. But I do like this. I like that their beards stretch down to show the age of these patriarchs. Of course, younger creationists do believe that these guys actually lived to that age, which kind of blows my mind. And I'm sure it blows yours too, but let's get into it. So Standing says, the Bible describes a time when people lived to over 900 years old. Yes, it does do that. Adam lived to be 930 years old, and this makes sense given the fact of genetic degradation. We're going to get into some genetic entropy conversation today. Now, for those of you who may not know, genetic entropy is a kind of New Age creationist hypothesis, if you will, proposed by John Sanford, who is, of course, a creationist. It's essentially a reskin of error catastrophe, which is an idea that in biology is pretty, it's not, like, accepted, you know? So I'm not sure why reskinning it was, you know, thought to be this great plan, other than you can now sell it to the masses who accept younger creationism. It relies on redefining what fitness is in an evolutionary context to go from something that's related to fitness being a reproductive output, how you're actually progeniting your genes to the next generation, passing them on, to being something called absolute fitness, which is nebulous at best. It lacks any concrete definition. You can't measure it. It's unempirical. It's basically like how fast, strong, or smart is the animal, right? It's like, okay, well, it depends. It depends on the environment. It depends on the organism. It depends on the context, the age, the ontogeny, a ton of different things. Ontogeny, age, kind of the same thing. But you get the point. Genetic entropy is not accepted by anyone at all. There is no genetic entropy consensus in biology that is positive for creationists. So I think it's funny that he says the fact of genetic entropy. Maybe in that reality, I suppose, in the creationist reality, but no, this is not something that is considered to be ubiquitous. If we take this point of most accumulating mutational damage back to a point of least accumulating mutational damage, that would be a time of perfection. So there's this idea that Adam lived to be 900, and people don't live to be 900 today because of genetic degradation. Essentially, genetic entropy relies on this concept of near-neutral deleterious mutations. It's slight mutations that act on the fitness of an organism that will accumulate and at some point cause a catastrophic extinction event. But over time, it's slowly building up to this point, making the organism worse, harming it in small ways. Of course, fitness acts on that, like selection for fitness rather acts on that, like all the time. We don't see any exception to this, except I think maybe it's been proposed for some viruses. I know Sanford has proposed it for H1N1, which was then BTFO'd, but let us continue. Genetic entropy, of course, being the thing we just mentioned, doesn't work. This would be a time of increased longevity. Adam and Eve would have had 0 mutations. Okay, just gonna let that one hang in the air. Then we have 2 nice kind of screenshots. One that says gene mutations began showing up in the last 5,000 years of human evolution, and another one that says protein coding mutations in people arose largely within the past 5,000 to 10,000 years. These 2 right here, of course, as you know, I always spring for the color copy. I always mention it because it cost me more and I'm bitter about it. It's kind of funny to me that, you know, neither of these, like, is the 6,000 year mark. Like, creationists tend to be like, yes, it's within the ballpark, you know, whereas conventional science will be like, looks like this is off a little bit. Maybe we should alter our hypothesis and, you know, look closely at the data and see what it tells us. But creationists will be like, if it's within 4,000 to 12,000 years, it indicates a young Earth. Okay. This idea, of course, that certain mutations, the majority of mutations that exist in a human population occurred like somewhat recently, given the mutation rate for humans, just isn't that surprising? Like, similar to creationists invoke the paper that came out 2 or 3, maybe 4 or 5 years ago, about how the majority of species that exist on Earth today arose 200,000 years ago. And that kind of makes sense. Species turnover is a thing, and I would imagine that with mutations, this is kind of a similar concept. They gave rise to humanity starting from a genome of no mutations. They were created perfect. As John Sanford points out in Genetic Entropy affirms Scripture, or as he points out, Genetic Entropy affirms Scripture, quote unquote. Genetic entropy was like hypothesized around Scripture, so that does make sense. It's that, he would hold that view. It's this idea of shoehorning it in, putting a square peg into a round hole. Of course, in this case, neither of those things really holds up. The veracity of Scripture throughout archaeological history or genetic entropy even being a thing, but I digress. We find generations in the ages of the patriarchs when they died in the book of Genesis. At the time of Noah, lifespans began to decline rapidly. Remember, the Bible claims to be history of the universe. I don't think it claims to be that. In fact, I've heard by most people that the Bible doesn't claim to be like it's a science book or history book or anything. It has strictly relationships to spirituality and your relationship with God or Jesus, right? Therefore, it should be accurate in all things science. Yeah, if it claimed to be a book of the universe, then yes, you would think that that would be the case. Creationists run into a lot of problems, though, with this, what they choose to be metaphorical and what they choose to be literal. For instance, they will look to Genesis 1 when it references the firmament, and they'll be like, the firmament is just like this separation of the heavenly realm and the earthly realm. The Hebrews thought the firmament was a solid thing. The word for firmament, rakia, is also used in like metallurgical senses almost exclusively, like throughout the book of Genesis, implying hardness, firmness, firmament, robustness, a barrier. So rakia isn't metaphorical. It's not meant to be metaphorical, which would mean, of course, okay, if you're going to take that stance that the Bible is a science book and must affirm science, then you've got a lot of explaining to do on where's the firmament at, what's being referred to there. The Bible gives us a perfect biological decay curve and shows extremely strong evidence that man has been degenerating from the very beginning of creation. No, it doesn't give us a perfect decay curve. There's like ages, I suppose, but there isn't like at age so-and-so genetic degradation was at this, and then at the subsequent age it was at this, and showing this nice curve. Standing is inferring that because ages used to be older, then there must have been a corroboration with that as far as ancient ages go, as far as like a best fit line. But the interesting thing is, there doesn't appear to be like a stark curve here. There doesn't appear to be anything exponential or linear, for that matter, going on with the patriarchs. It's just like all of them are kind of old, I guess. And then after Noah, everyone starts dying really young. There isn't like a slope there. It's like clunk, plummet. So, again, I don't know that I would call that a decay curve. Critics of biblical creation will say Adam and Eve would have committed incest and inbreeding, and it would have been a major problem. So Adam and Eve are false. And that's how I say it. I don't know about you, but there's a lot of weird stuff with that, right? Like, it kind of blows my mind that people don't harp enough when arguing with creationists about how in, like, Leviticus, God's like, hey, don't do the incest. And then, like, 2 times in the history of the world was like, hurry, do the incest. There aren't enough people. So that seems a little bit against God's nature. You like reading what would it be called a proper exegesis of the scripture feels kind of against God's nature. So either God's nature is wrong, or we're interpreting those stories wrong in that we're looking at them in a literal sense. I'm agnostic, and I don't even think that you should interpret those literally. I think that those are metaphorical, or were at least understood by the Hebrews to be phenomenological, like almost like a folk tale, if that makes sense. But it doesn't matter either way, whether they thought it was literal or not, it still didn't happen, that being 6,000-year-old creation in a flood. It is true that we cannot have all human beings descending from just two people, because we would immediately, is it true? That's right, I thought that seemed a little bit lucid for saying. Is it true that we cannot have all human beings descending from just two people because we would immediately be having these serious inbreeding problems? Inbreeding exposes the hidden reservoir of genetic mistakes. If there are no mutations, there are no deleterious effects from inbreeding. This would not have been a genetic problem because there were no mutations at creation. So that's part of the problem, but the other part is like having genetic diversity to prep a population, there is no forethought obviously, but to have a well-prepared population in the event of disease down the line, or having an available genetic pool to select for certain traits if the environment changes, or things like that. Yes, inbreeding is certainly a problem, but not in the sense that he's kind of going for here. He's not having an all-encompassing understanding, or rather, he's not displaying an all-encompassing understanding of the problems with inbreeding. I always find it funny when uninformed evolutionists will say, we have an inbreeding problem, when in fact, it is the evolutionists who have the inbreeding problem. I discuss this in detail in my previous book, Universal or Separate Ancestry, the Biblical Model of Origins Made Easy. On page 66 and 67, I point out, and then he has like this massive section of this book that is just like a copy-paste from the other book, which is kind of, I don't know. I like, you could have maybe paraphrased it, or whatever. I think it's funny to kind of quote yourself. But let us continue. It is also important to note that since creationists invoke created heterozygosity, there would be no inbreeding problem after creation and after the flood. The creation model's bottlenecks were all benign and only one generation. The bottlenecks were benign. Okay. Let us pause for a moment and appreciate the invocation of created heterozygosity. This is the idea that organisms were created with this sort of pre-programmed diversity to prevent the problems that I mentioned moments ago, with inbreeding down the line and a lack of diversity to pull from in the case of environmental change or catastrophe or whatever. But excuse me. But this created heterozygosity thing becomes very problematic, specifically when we look at the genomes, particularly large genomes of some organisms today. Chiropterans, for instance, so the bats, would have genomes just thousands upon thousands of times greater than they have today in order to explain the diversity that we see in the massive diversity of chiropterans. And that genome of that progenitor pair would be biologically impossible to have at all, right? Like this genome is just massive. It is mind-blowingly large. So, it's the little things like that add up and become problematic for this bottleneck nonsense. But let's continue. They were all followed by rapid exponential population growth, which would guarantee limited loss of created heterozygosity. I don't think that is the case. This rapid exponential population growth, like, I'd like to see some math for that, because I've seen AIG present, Answers in Genesis, the Creationist organization, present two primary population growth curves, where they essentially say, look, after the flood, we would have had plenty of people within the first 200, 300 years to make the pyramids of Giza. Don't worry about it. It's all fine. And of course, there are many problems with that as a concept, this idea from going from 8 people to hundreds of thousands in order to explain the appearance of megalithic structures like the pyramids of Giza, but also all of the structures prior to the pyramids of Giza, like the Red Pyramid, the Bent Pyramid, and all of those mastabas and necropolises. And that's just in Egypt. But the growth curves that you're having to invoke in order to get enough people to create these things, had they been sustained, which AIG or Standing for Truth give no reason to halt that level of population growth, to say, okay, it goes to this point and then it stops and it levels off and it becomes a normal population growth. We would have trillions of people on the planet by now, which, of course, we don't. We don't have that amount of people. So obviously, either the growth rate leveled off at some point that AIG and Standing invoke, or alternatively, it was never there in the first place. So problems abound. Also, by the way, those growth curves don't take into account infant mortality, famine, pathology, things that are just going to chip away at the human growth rate back in the day, because we didn't have medicine or proper care. The evolution model, on the other hand, assumes all DNA differences are the result of mutations and genetic mistakes over time. Therefore, our hypothetical population bottleneck in the out of Africa hypothesis is not even remotely possible. Humans at this time would have already accumulated enormous amounts of genetic mistakes. There is no source for this. There is no math for this. Standing has, in my opinion, a very rudimentary understanding of how mutation and selection operates. They also invoke this population bottleneck to explain away the low genetic diversity found in humans. They need a way to reduce homogeneity, and they do so by invoking this bottleneck roughly 70,000 years ago. This bottleneck apparently reduced the human population at the time to approximately 2,000 to 10,000 individuals. And then we get a screenshot of Wikipedia right there. Boom. And it talks about the genetic bottleneck theory. Let us take a stroll through the literature very briefly and see what we find about this genetic. So for those of you who maybe don't know, Standing for Truth has been corrected on this idea that a population bottleneck is universally accepted by anthropologists, by the likes of CRISPR and others, because it's not. And this is a paper from 2018 that kind of gets into why the Toba eruption as this reason for low genetic diversity in humans is pretty falsified at this point. Why it's not accepted anymore, but it wasn't even universally accepted then either. This is, again, 2018 paper. Now, something else that I want to clarify for Standing for Truth is, again, I have an inkling that he probably watches these videos because they concern his book, is that human genetic diversity, as I've pointed out before, is not like anomalous among animal populations. We aren't even the least diverse within the hominoids, let alone in general, in the animal kingdom. I've shown Standing a video before, or rather in one of my videos before, a quote from a paper that was essentially like, yeah, we fit quite well with the rest of the animals as far as our genetic diversity. It's on the lower end, yes, but it's not, again, anomalously so. So let's look into why the Toba extinction, the Toba super eruption, is not credited for low genetic diversity. And then we'll discuss very briefly why humans do have low genetic diversity. So this paper is titled Subdecadal Phytolith and Charcoal Remains from Lake Malawi in East Africa Imply Minimal Effects on Human Evolution from the 74,000-year-old Toba super eruption. So, from the abstract, the temporal proximity of the 74,000 Toba super eruption to a putative 100 to 50,000 human population bottleneck is the basis for the volcanic winter/weak Garden of Eden hypothesis, which states that the eruption caused a 6-year global volcanic winter and reduced the effective population of anatomically modern humans to fewer than 10,000 individuals. To test this hypothesis, we sampled 2 cores collected from Lake Malawi with cryptotephra previously fingerprinted to the Toba super eruption. Phytolith and charcoal samples were continuously collected at a 3 to 4 millimeter or 8 to 9 year intervals above and below the Toba cryptotephra position, with no stratigraphic breaks. For samples synchronous or proximal to the Toba interval, we found no change in low elevation tree cover or in cool climate C3 and warm season C4 xerophytic and mesophytic grass abundance that is outside of the normal variability. So pause right there and appreciate what's being said here. So they took soil samples or rock samples from above and below the Toba super eruption, mark the signal, and they found that there wasn't a decrease in the variety of different kinds of plants that were thriving in the area, implying that since plants are the most vulnerable to volcanic winters, things were pretty okay in East Africa at this time period. So let us continue. A spike in locally derived charcoal and xerophytic C4 grasses immediately after the Toba eruption indicates reduced precipitation and die-off of at least some Afromontane vegetation, but does not signal volcanic winter conditions. A review of Toba-Tuff petrological and melt inclusion studies suggests a Tambora-like 50 to 100 MT, I think that's sulfur oxate, SO2 atmospheric injection. However, most TOBA climate models use SO2 values that are 1 to 2 orders of magnitude higher, thereby significantly overestimating the amount of cooling. A review of recent genetic studies, this is key standing, finds no support for a genetic bottleneck at or near 74,000 years ago. Based off of these previous studies and our new paleoenvironmental data, we find no support for the Toba catastrophe hypothesis and conclude that the Toba supereruption did not, 1, produce a 6-year-long volcanic winter in eastern Africa, 2, cause a genetic bottleneck among African anatomically modern human populations, or 3, bring humanity to the brink of extinction. So this is something it's kind of important to appreciate. And, this is the point where usually I would say, let's not just read the abstract, let's get into the methodology. But I don't think that I understand geology well enough to be able to kind of parse out the importance within the methodology. So I'm going to leave this at the abstract for you, and leave a link in the description if you would like to go and investigate a bit further into the paper. Plus, we're going to be going over, you can see from the top, we're going to be going over some other papers. So the significance of this is essentially that Standing says human genetic diversity is low, and the only explanation for this is a global flood. Evolutionists, on the other hand, notice that human diversity is low and have to invoke an imaginary bottleneck in order to explain it. We don't accept that bottleneck occurred because there's no support for it, paleoenvironmentally speaking, or genetically speaking. Now, there have been bottlenecks in human history, but they're not bottlenecks in the sense of, in an ecological sense, in that there is something that is taking out a lot of the original population, leaving few to reproduce. These bottlenecks, conventionally speaking, this low genetic diversity is caused by the founder effect, human populations leaving Africa in small bands, tribes, things of that nature, and having little genetic diversity in those local pools as they spread out and kind of colonize new areas. This is well understood. In fact, this is what's also thought to have occurred with the Neanderthals and why their northernmost populations had such low genetic diversity. And again, human genetic diversity just also isn't that low when compared to, especially the other great apes. So let us continue with Standing's book here, and I'm going to stay in small mode because I think we move on to our next paper here soon. This bottleneck was not 1 or 2 generations. It apparently lasted for roughly 1,000 years. Again, no. This means that the human population would have been incredibly compromised genetically. Again, no. These recessive mutations that have been accumulated over their deep time evolutionary history would then come to the forefront and lead to rapid genetic degradation. That sentence is an affront, honestly. So this idea, right, is essentially that Standing proposes that evolutionists would have a problem with genetic entropy, something we don't even see any support for and thus don't accept. No. The next time a critic wants to accuse biblical creationists of having an inbreeding problem during the early parts of human history, make sure you point out that it is the evolutionary model that suffers from an inbreeding problem. So let us appreciate the comparison here. And we'll do so on his terms, despite the fact that, remember, we're not actually working with a 75,000 year ago bottleneck, and that human genetic diversity is just not all that surprising. But so Standing has this idea, this comparison in mind, where he says people who accept evolution have a low genetic diversity in modern human populations, and they have a genetic bottleneck that they must invoke 75,000 years ago, and those two things in tandem, 1, don't work because they're a rescuing device, and 2, don't work because genetic entropy, something they don't even accept, would act on those populations and cause error catastrophe, but the reskin version, so the genetic, tailspin, essentially. Then he says, young Earth creationists don't have that problem. Because even though we're starting from 2 people instead of 10,000 individuals, God front-loaded the genetic diversity into the original progenitor pair of all humans. Therefore, they don't have a genetic diversity problem, and also they can breed very quickly after these mass extinction events in the case of Noah's flood, for instance, and therefore we can get both the populations and the diversity. Both of those things, the population growth curve and the created heterozygosity, have no empirical basis. There is nothing there that is even remotely measurable. It's an idea. created heterozygosity is an idea. And it's one that is falsifiable. Because, again, if we look to the chiropterans, the bats, we see their vast diversity today. And in order to have a single progenitor pair of two bats, the bat kind, leading to all of the bats that we see today, their genome is physically impossible size-wise of that progenitor bat pair in order to see. So, again, there's not just no empirical support for created heterozygosity. There is indeed support against it. So we're not dealing with the top brass hypothesis here. Of course, this comes from Nathaniel Jeanson, who we'll be dealing with momentarily. All right, let us continue. All we have to do is look to the cheetahs in the world today. Oh, honey, you don't want to talk about the cheetahs. They have experienced a population bottleneck and are at risk for extinction. They are down to roughly 7,000, and they are experiencing rapid genetic deterioration due to inbreeding. Should we expect this population of 7,000 cheetahs to spread out in all parts of the world, seizing dominion of the planet? No, of course not. And yet this is exactly what proponents of human evolution believe would happen in their fairy tale out of Africa population bottleneck scenario. This close to going and getting a glass of wine after that one. So there are a couple of big time problems here. Let us, I know I say let us a lot, but let's appreciate this for a moment. First we have the cheetahs. And Standing notes that they're down to 7,000 are experiencing rapid genetic deterioration due to inbreeding. Now I thought that they were front-loaded. I thought they had front-loaded heterozygosity so that they could withstand being down to low numbers. What happened to the heterozygosity? What does it wear out? This is what I'm saying when I'm saying there's nothing empirical about it. What is the mechanism behind created heterozygosity? How does it behave? How do you get down to 7,000 in the cheetahs and have a problem, but with the Noah's 8 or the original 2 humans, no such problem exists. This is the case with many animals today that are experiencing low genetic diversity, and with cheetahs, they're almost genetic twins to one another, but that's how bad of a population bottleneck that they've experienced. How does this work? What is the model here with the created heterozygosity? Should we expect this population of 7,000 cheetahs to suddenly spread out into all parts of the world, seizing dominion of the planet? That's insane. Like, the idea that low genetic diversity would be what prevents humans from being able to go out and migrate and colonize and settle new locations is insane. But the comparison to the cheetahs is even more bonkers. Cheetahs don't do that. Humans do that. The ancestral populations, the hominins of humans, are notorious for doing. They spread all around Africa first, and then they were leaving as early as Homo habilis, which is, we're very curious. We have large brain case sizes, the largest brain-to-body ratio with regard to the surface area and things like that in many ways of any of the primates indeed, but most mammals too. So this idea that low genetic diversity would be, like the humans are like, damn, like we've got really low genetic diversity, I guess we better not spread out, or that would be encumbering to them in some way is silly and nonsensical. Okay. Then we've got another cribbed, if you can see that. Let me see, hold on. Let me make sure that you can, actually, yeah, there you go. Some cribbed stuff there, nice. The evidence is clearly in line with independent origins. Universal ancestry has been thoroughly refuted. On page 49 to 50 of my book, Universal or Separate Ancestry, the Biblical Model of Origins Made Easy, I demonstrate that the creationists are the ones making the predictions. And then we get another, I don't even know how many pages this is, like a copy-paste from it goes from this page to the next page and ends over here, just like cribbed from his other book. Technically, you're not even allowed to plagiarize yourself in academia. But speaking of academia, universal ancestry has been thoroughly refuted. I look forward to the manuscript, and I've said this before, but why is he publishing books on Amazon? Like, why not just publish a paper? Raw Matt tried, but we all know how that turned out, don't we? Proponents of evolution and critics biblical creation are now faced with many extremely serious challenges. One of these challenges involves pedigree-based human mitochondrial DNA mutation studies. And then he shows a screenshot of the pedigree-based thing from, looks like Wikipedia again. The reason for this is because pedigree-based studies and the empirical method rejects deep time evolution and universal common ancestry. No, we're going to talk about this in a sec. The best tools to determine what is related and what is not related are molecular clocks and DNA function. Oh, you do not want to be saying that, my guy. Trust me, you don't. These are the lines of differentiating evidence that can help us answer the questions of ancestry once and for all. There are many indirect lines of evidence that can help give us clues into the question of ancestry, but the only direct way is genetics. This is what is inherited, sperm and egg. Molecular clock data in both the mitochondrial DNA and Y chromosome confirm a young Earth timescale and refute an old origin of species. And then I've put a question mark next to the Ann Gibbons, the Ann Gibbons screen grab. I've talked about Ann Gibbons before. Again, this is not an appropriate use of sourcing. How do apologists of evolution explain away data? Evolutionists love their rescuing devices. Evolutionists look to what is called time dependency to explain away the data. They have suggested that the mitochondrial DNA clock ticks at different rates in different points in history. Is this a good explanation? I do like this. Is this a good explanation? I like that. We're going to finish this next paragraph and then we'll dive into this section. Evolutionists would have us believe that mutation rates have been significantly slower in the past. Should we accept this explanation? For this explanation to be scientific or even reasonable, scientists of evolution must make testable predictions. Dr. Nathaniel Jeanson has made significant testable predictions on mutation rates. He has taken the observed mutation rate in non-African people groups and made future testable predictions on African people groups whose mutation rates are not yet known and have not been measured. During a debate with Dr. Dennis Venema of Biologos, he was associated with Biologos at the time, Dr. Venema admitted he could not make the types of predictions that Dr. Nathaniel Jeanson was making. The winner of the creation versus evolution debate will be decided on who is making the best testable predictions. So, your testable predictions, they need to be accurate, right? That's the first thing that I want to kind of touch on there. And Nathaniel Jeanson does not make accurate testable predictions, regardless of what you may have heard. I went over some of his Y chromosomal work in one of my previous videos, link in the description if I remember. But this is not something that he's quite good at. Nathaniel Jeanson is very good at saying, here are my predictions, now someone else test them. For example, that mention of the testable predictions on African people groups whose mutation rates are not yet known, that's a reference to the Khoisan people. That prediction was made years ago, and Jeanson hasn't done the legwork for it, right? I don't understand why. If it's so easy, it would be so easy to vindicate your hypothesis, you know? But let's get into some of this. Let's see. Pedigree-based mutation rates. We're going to talk about that really fast. And in a little bit, I believe we'll go ahead and discuss Jeanson too, and Y chromosomal stuff. Well, we're just gonna bop through a couple of papers real quick. It's going to be fun. So first, let's talk about pedigree mutation rates. Now, something Standing has been again corrected on before that I suppose he just isn't implementing into his knowledge base, I guess, is that pedigree rates are necessarily faster than phylogenetic rates. He's had this explained to him by CRISPR, he's had it explained to him by Dan, some others I believe as well, and he follows the logic. He'll follow the logic with you down that rabbit trail, but for whatever reason, this you reach the conclusion it's like 50 first dates, and then all of a sudden he forgets the concepts behind it, the reasons why these phylogenetic rates are necessarily going to be slower than pedigree rates. And we'll talk about that in a little bit too. But I think that this is a great paper to discuss very briefly. Oops, not this one. This one. Direct estimation of mutations in great apes reveals significant recent human slowdown in the yearly mutation rate. So the interesting thing about this paper is that what Standing had said just moments ago, where he discusses how pedigree rates are essentially the superior method. The reason for this is because pedigree-based pedigree-based studies and empirical methods reject deep time evolution and universal common ancestry. And what this paper did is it used direct pedigree mutation rates for humans, gorillas, orangutans, and chimpanzees, and found that those pedigree rates backed up the fossil record. Boy, that's really weird, isn't it? So let's read the abstract. The human mutation rate per generation estimated from trio sequencing has revealed an almost linear relationship with the age of the father and the age of the mother. The yearly trio-based mutation rate estimate of 0.43 times 10 raised negative 9 is markedly lower than the prior indirect estimates of 1 times 10 raised negative 9 per year from the phylogenetic comparisons of the great apes. This suggests either a slowdown over the past 10 million years or an inaccurate interpretation of the fossil record. So that's the problem that they've presented. In an abstract, you present the problem, and then you show how your work is potentially the solution. So here we use sequencing of chimpanzee, gorilla, and orangutan trios and find that each species has a higher estimated mutation rate per year by factors of 1.67 plus or minus 0.22, 1.54 plus or minus 0.2, and 1.84 plus or minus 0.19, respectively. These estimates suggest a very recent and appreciable slowdown in human mutation rate, and, if extrapolated over great ape phylogeny, yields divergence estimates much more in line with the fossil record and biogeography. So that hypothesis of the hominin slowdown hypothesis, the hypothesis of the hominin slowdown, the idea that these mutation rates in the human lineage have been slowing down over time, has been in existence for a much longer time than this paper. And now this paper is coming to fruition. It's showing that is indeed heavily supported when we look at the other great apes, which is interesting because I thought evolution didn't make testable predictions. Evolution makes testable predictions in pretty much every ecological paper, period, especially if you have to run phylogenetic corrections, which I did in my own thesis. So, it kind of betrays a lack of literature work done by Standing if he thinks that these predictions aren't panning out. But let's continue. So this paper is very interesting. They went ahead and looked at the mutation rates for the pedigree mutation rates for trios for the great apes. They didn't do bonobos, but they did do pans, so common chimps. So it does indeed cover their bases. And we've got this lovely graph right here, or lovely figure right here. Estimates of great ape divergence, genomic divergence time and species separation times. The estimated mutation rates per year for each species extrapolated over the phylogeny yield divergence time estimates, blue numbers, which are much more in line than estimates based solely on the current estimate of human mutation rates, green numbers. So what we see here is that when accounting for mutation rates for each species and the hominin slowdown hypothesis, we match up with the fossil record for divergence times. Who would have thought? That data doesn't make sense at a younger time scale, does it? They continue to talk about their methodology, how they've done this, samples and methods, sequencing, things like that. But I want to get down to the discussion, if we can, or just the conclusion, because it's interesting. Okay, phylogenetic dating. The great ape phylogeny deviates slightly from a molecular clock, according to Murjani et al., who we're going to be looking at. I believe she does this. Yeah, Murjani et al., Priya Murjani, which we'll talk about in a moment. With the chimpanzee branch being 2% longer than the human branch, the gorilla branch 6% longer, and the orangutans 11% longer than the human branch since their common ancestry. Using these numbers, we focused on the human branch. The branch links from human to common ancestor with chimpanzee becomes 0.006, gorilla 0.008, and orangutans 0.01. So the funny thing is that they were mostly in line anyways. And when accounting for the slowdown, you get these incredibly small numbers as far as a difference in what is seen in the fossil record versus the genetics. Using estimated chimp mutation rates since the common ancestry between humans and chimps, this corresponds to 9.46 million years for the human chimp average genomic divergence time. Using gorilla rate 0.66 since the common ancestry between humans and gorillas becomes 12.42 for the human gorilla average genomic divergence time. Using orangutan rate 0.00079 since their common ancestry becomes 21.85 million years for the average human orangutan genomic divergence times. To turn the divergence numbers into estimates for species separation, we used the ancestral effective population size reported by Scally et al., scaled to the mutation rates assumed in common ancestors, yielding the human-chimp effective population sizes, gorilla and orangutan, and you get the following estimates. About 6 million years for human chimp, about 8 million years for human gorilla, and about 15.49 million years for orangutans, which matches up with the fossil record. So this is this is really damning as a paper for young Earth creationism. You have a previous hypothesis that is tested using what creationists deemed to be the appropriate way of testing for mutation rate, which is the pedigree-based mutation rate. You account for phylogeny, you back it up, and what do you get? You get a corroboration of the fossil record for human evolution. So that's kind of cool. Also, the evolution of mutation rate across primates is very interesting because in this paper it talks about not just the slowdown rate for hominins, but also for other primates, which other primates have indeed are showing support for a slowdown in their mutation rates. Specifically, I believe this is in some of our prosimians. So that's kind of cool too. We'll go down to the conclusions and future directions. Recent studies have shown that both the mutation rate and spectrum evolve rapidly across primates. Converging evidence from pedigree, phylogenetic, and fossil comparisons supports a mutation slowdown rate in humans and possibly other primates. The magnitude and timing of the slowdown, however, remains uncertain. Now, the cool thing is that this paper, if memory serves, let's see, where's our date? Show me the date. Show it to me now. This is a 2020 paper, so this is subsequent to this direct mutation paper over here. So it's able to incorporate it in, and I believe probably cites it. Let's see, who's this by? This is Bessenbacher et al. From 2018. Let's check it out just out of curiosity. Bessenbacher, let's see. Okay, what kind of, are they doing this by like, well, we'll just control F. Bessenbacher, right there. Bessenbacher. Boom. There it is again. Again. And show us in the, direct death. There it is. Yeah. So that information is incorporated in here. And oh no, what happened? Let's click back. Show me the money. There it is. Nice. So back to the conclusion. Okay, the magnitude and timing of the slowdown remains uncertain. This has major implications for the use of mutation clock for dating evolutionary events and hence for our understanding of the chronology of primate evolution. The fact that different types of mutations occur at different rates across primates and depend on distinct biochemical and life history factors highlights that there is not just one but many molecular clocks each ticking at a distinct pace across primates. Thus, a way forward is to consider each mutation type or mutation signature separately and characterize the rates and factors separately. That impact its evolution. For recent timescales, the use of securely dated ancient DNA specimens may also provide a way to generate time-dependent estimates of mutation rates over different evolutionary periods that can be used to recalibrate their molecular clocks. New insights into the genesis and evolution of mutation rate have uncovered gaps in our understanding of the mutation process itself. If DNA damage and not replication errors drive the variation in mutation rate, how then to explain the correlation in yearly mutation rates and generation times? One possibility is that these differences instead reflect the changes of DNA damage due to other life history traits like longevity, metabolic rate, or biochemical factors that co-vary with generation time. Differentiating between these hypotheses require new models to disentangle the correlation of life history traits as well as new comparative data to test the robustness of these inferences. So, cool. That's pretty classic. Finally, as DNM data from more species and larger sample sizes become available, it is important to set reliable standards for evidence analysis and filters so that the mutation patterns can be readily compared across studies. Investments in generating better and more complete assemblies, particularly without reference bias, will be timely. The application of newer technologies with single-cell sequencing, such as single-cell sequencing, will be highly informative for directly assessing the variability in germ cell mutation rates. Together, these efforts will improve our understanding of the process of mutagenesis and evolution across species and help reliably set the molecular clocks for reconstructing the timeline of primate evolution. So the cool thing about this is that, yes, we know that mutation rates change. We know that humans have experienced a slowdown, and so probably have other primates. So this is not like a rescuing device. This is something that is empirically supported, unlike the idea of created heterozygosity or random population explosions and then plateaus. It doesn't follow. None of this follows. So let me make sure that we've we got to talk about Nathaniel Jeanson too. Then he talks about how Darwin knew nothing about genetics. So again, the whole point that just to round things off, the reason I brought that up is to show that pedigree studies, they corroborate human evolution. They corroborate deep time in evolutionary theory. And for standing to say that empirical pedigree-based studies reject evolution and common ancestry belies a absolute deficit in understanding of what pedigree-based studies have been done within humans and within other primates. So he talks about how Darwin didn't know about genetics, blah, blah, blah. Or is time dependency a rescue device that evolutionists are forced to invoke to explain away observable data? This is observable data. Created heterozygosity is not. There's a very, very large chasm between those two. Once again, the evidence is clear. We have traces of Adam and Eve in our genetics. The mitochondrial DNA takes us right back to one female ancestor just thousands of years ago, and this conclusion is based on pedigree mutation rate studies. Then he shows us Parsons, and he shows us Douglas T. Rode, modeling the recent common ancestry of all living humans. Experts in the fields related to evolution and uniformitarianism struggle with the fact that mitochondrial DNA clocks point to a young creation. No, mitochondrial rates do not. I have talked about the problem with Parsons, the invocation of Parsons et al. First of all, it's funny that they have 6,500 years here, 6,500 years here for the last common ancestor, mitochondrial Eve, for humans using the Parsons paper, because the Parsons paper has a massive error bar range. It's something like she could have lived from 1,000 years ago to like 1.2 million or something like that. I say it specifically in the conversation I had with CRISPR, which I will link in the description. It's actually just from the I'm almost done with these, I think I'm done with these creationists video. But I'm so eager to talk about genes then. What testable predictions regarding molecular data can evolutionary scientists make? You mean like the ones we just went over? The critics of biblical creation model have failed to make any reasonable or compelling argument. The gold standard of science is to make testable predictions that are falsifiable. So how would you falsify created heterozygosity? How would you falsify that? I've yet to hear that from these guys. Do critics have a better explanation for the mutation rate data we have for humans and certain species? Yes, what I just mentioned. Why can they not make testable predictions? What I just mentioned. It is the biblical creationists that are making predictions and confirming their model. We've seen earlier in my he references the other book again. I even put again in the side chat, in the sidebar. It is not just humans he claims to make predictions on, he also does it with other species. This is a reference to Jeanson's book, Replacing Darwin. And of the 7 species he looks at, I think like 3 he claims match his work, and 4 don't. So I guess there's some welcome transparency there. He welcomes the challenge and is more than willing to test the predictions in the lab. Then why hasn't he done that with the Khoisan people or other, testable predictions he's made that he has not tested himself? Sometimes they say substitution rates will save them, and sometimes they say natural selection will save them. They insist mutation rates must have been slower in the past. They were, just showed why. Why? Because, of course, evolution happened. There is no questioning the basic assumption or premise. In their minds, evolution has occurred, end of story. Kind of, yeah. I mean, kind of. The empirical data supports that. There's nothing that supports a young Earth in any field, actually. I've said that before. I realize maybe that's a little intense, but it is the case. Regardless of the rescue device, it cannot lead to testable predictions, then it is not science. It is actually pseudoscience. Oh, the self-werewolves vibes here. What do we know? We know the empirical method confirms about what he's just repeating the same thing. Then he complains about the Y chromosome. He says that genes is making predictions on the Y chromosome. I've explained in the past why you can't use the Y chromosome to predict populations of the past, because it's coming down the dad's line, right? Like you can't, what if you have no sons? It doesn't work. This isn't how any of this works. Then he goes, they will say, we need to calibrate the observed mutation rate within their deep time evolutionary history. They want us to calibrate the empirically derived mutation rate with their assumed geologic column. Does this make any sense? And then he has another screen grab that says, well-designed pedigree studies may be more reliable than phylogenetic approaches. This is why I said that these papers that I just mentioned bode so poorly for standing for truth and for creationists in general. The deep time assumption is exactly what is in question. It's not, the heat problem, the geologic column, the deposition rate of pretty much every kind of sediment. It's bad news all across the board, the way that the tectonic plates move. There's nothing in geology that is even close to being sympathetic towards creationism, young Earth creationism, let alone supporting it. I know I've reiterated this over and over again, but to the point where it may sound redundant, but it is really of the utmost importance that the evolutionary community steps up if they have any chance at putting forth a better model. If the evolutionary community thinks they can match up the biblical creation model of ancestry, they need to make better testable predictions. I put okay SFT because it just, the absolute dissonance here blows my freaking mind. I can't believe the attitude that's being put forward here. It again, and I know I've said this to the point where it may sound redundant, but it belies such a deficit in literature, like understanding and legwork, for lack of a better term, right? Like he doesn't understand that if it's an ecology paper, there's like evolution is necessarily involved. If it's a biology paper, it's almost necessarily involved, and that's why Theodore Dobzhansky said nothing in biology makes sense except in light of evolution. It's a linchpin to the entire field. He really doesn't want to go down the geology route. Certainly not. I'm just making sure we get all our genes. Yeah, we've got more genes and stuff. That's why I'm holding off. I'm charging up the punch. Then he talks more about Y chromosomes. He gets upset that at some point here, I think it's on the next page, that human and chimp Y chromosomes are very different from one another. Let's talk about that really quickly. I've done this before already, but the interesting bit about Y chromosomes is that of the great apes, the chimpanzee and bonobo Y chromosome is the most divergent. So humans and gorillas share more structurally. In identity, we're still more similar to genus Pan, which identity is the important bit. It's actually what is encoded in the genome. But let's appreciate this. Monica Shakova. I reached out to her on Twitter, by the way. Okay. The mammalian male-specific Y chromosome plays a critical role in sex determination and male fertility. However, because of its repetitive and haploid nature, it is frequently absent from genome assemblies and remains enigmatic. The Y chromosome of great apes represents a particular puzzle. Their gene content is more similar between human and gorilla than human and chimpanzee, even though human and chimpanzee share a more recent common ancestor. To solve this puzzle, we've constructed a dataset including Ys from all extant great ape genera. We generated assemblies of bonobo and orangutan Ys from short and long sequencing reads and aligned them with the publicly available human, chimpanzee, and gorilla Y assemblies. Analyzing this dataset, we found that the genus Pan, which includes chimps and bonobos, experienced an accelerated substitution rate. Pan also exhibited elevated gene death rates. These observations were consistent with high levels of sperm competition in Pan. Furthermore, we inferred that the great ape common ancestor already possessed multi-copy sequences homologous to most human and chimpanzee palindromes. Nonetheless, each species also acquired distinct ampliconic sequences. We detected increased chromatin contacts between and within palindromes, likely facilitating gene conversion and structural rearrangements. Our results highlight the dynamic mode of Y chromosome evolution and open avenues for studies of male-specific dispersal in endangered great ape species. So it's a cool study. It was published in PNAS. Let's see. I want to find some of our... I know there's some pretty interesting... Here we go. The nucleotide sequences here. So when we're looking at nucleotide sequences, regardless of how you hash it out, even though, like I said earlier, there's more structural similarities between humans and gorillas because genus Pan is the one that's diverged from the rest, sequence-wise, it's still humans and genus Pan, and sequence is, again, what is vital. So we've got Y chromosome here, over here on the left, and then over here on the right, we've got a full autosomal genome. Or just autosomes. They talk about content evolution. This is another one that's really important because this is the gene content. So this is the actual makeup. And you still, identity rather, you still end up with humans, with chimps and bonobos. I mean, this is a very clear paper that I think is highly problematic. Here's another one here discussing specifically palindromes between humans and chimps. And let's get down to our discussion, substitution rates. Show me the money, future directions, and they just talk about the content. Let's see, identity. We'll control F. That's how real science works. All right, this is what we wanted, the alignments. Okay, for example, as was shown previously, the gorilla Y had the highest percentage of a sequence aligning to the human Y in terms of sequence identity. The chimpanzee and boy were most similar to each other. Pairwise alignments, lowest to any multi-sequence. For multi-species alignments, the human Y was most similar in sequence to chimpanzee and bonobo Ys, less similar to the gorilla, and least similar to the orangutan Y. So this is what I was talking about. So when you align them up, so the percentage of sequence aligning structure, you get most similar to gorilla, because again, chimps and bonobos are most divergent. But for content in sequence, most similar to chimps and bonobos, 97.9 and 97.8 respectively, 97.2 for gorilla. So we're gonna shut the case on the Y chromosome stuff. Standing's got to explain. He's got to explain why that doesn't hash out, and also why these similarities are so similar if humans are not a part of the ape kind. Scientific data does not lie, true, and modern scientific data tells us all men have similar Y chromosomes. He's still talking about that. Then he goes, we can find traces of Babel in our genetics. No. Then he talks about more for the haplogroups. He shows the unrooted tree. I put unrooted, really? Now, fortunately, Standing has, at least in my chat, admitted that he does not know why this unrooted tree is utilized here, and why Jeanson hasn't rooted it. What's going on there? He also says evolutionists are losing the war, and they know it. Let's see. We've talked about mitochondria. Well, we're going to finish talking about mitochondria in a second. But we've talked about pedigrees. We've talked about the Y chromosome. Let's see here. He talks more about the chimpanzee. He also uses Jennifer Hughes' paper for chimpanzees and human Y chromosomes are remarkably divergent in structure and gene content. And then he uses the screenshot here. But despite the fact that this paper, or rather that this paper, God, I wish, that this book was published last year, in July 2020, he has not included the direct estimation of great ape pedigrees, which was from 2018 in the paper. Nor has he included this one, which was, I believe, January, maybe? Whatever. The point I'm making is that there's no excuse for including Jennifer Hughes from like 2016 and not including these two. Unless you had some kind of agenda. I don't know. Maybe that's crazy. So then he talks about taxonomically restricted essential genes, which is what separates humans from the other apes. He doesn't list what taxonomically restricted genes those are, and he also doesn't talk about how when we're looking at taxonomically restricted genes, humans and chimps share more of those taxonomically restricted genes than they do with any other animal on the planet, and chimps share more taxonomically restricted genes with humans than they do with any other animal on the planet. Again, landing them together. So you don't want to use, because you don't want to use this as your separation, or else you're going to separate chimps from bonobos in the same way. One final thing before we move on. Okay, then he talks about ENCODE. So ENCODE is another biggie. Standing for Truth thinks that the entire human genome is functional because of the work of ENCODE, whose definition for functionality was simply that it was transcribed. And they said 80% of the human genome is functional. Curious that that's not 100% because it's transcribed. Cool. But then most of the people who worked on ENCODE actually walked that back later and were like, yeah, our definition for functionality was really bad. Dan Graer also did some work where he showed that the maximum bound for functionality in the human genome is probably going to hover around 25%. Part of the reason we know this empirically is because for other organisms, like, say, mice, you can run knockout tests. And I've yet to hear Standing discuss the knockout tests and why they're not a valid means by which we can assess functionality. Because you can take a mouse and knock out over 50% of its genome. Knocking it out, meaning in vitro, you're actually clunking it, you're destroying it for all intents and purposes. And that mouse can move on, be born, and have babies that are also fine with half of its genome destroyed because it's non-functional, it's not doing anything important. And the same is true for humans, in that most of the genome doesn't do anything. So, I don't know, I've yet to see any responses to, how those knockout tests hash out, how the ENCODE researchers have problems with their own definition. Graer's empirical work with the limits of human functionality in the genome. So with that, I think we've covered everything. We've covered ENCODE, whatever, which means we can move on to talking about Jeanson very briefly. But let's read his kind of last paragraph here. He says, imagine a gene that tells a gene, which in turn tells another gene what to make and when to make it. In a genome that is functional, it's frightening to think about all the things that could go wrong. He's basically like, genome complex, guess it was designed. There's an obvious shelf life on genomes of living organisms. The fact that we're still here is 100% proof that we've not been here that long. Low-impact mutations continuously accumulate in the genomes of living organisms and slowly degenerate these amazing complex information systems. This is all evidence of one perfect creation going downhill. As John Sanford points out, we are a dying people in a dying world. Our hope is not in this world. Our hope is with God in heaven. Our hope is in Christ. Again, genetic entropy, there is no support for it. You have to change the definition of fitness. And Standing will say, because she says we have to change the definition of fitness, that means she's not been reading or listening to my content on it. It's not that simple. It is. I've heard you talk about how alternative fitness or ultimate fitness is a better means of measuring it, of measuring an organism's kind of measuring the organism's fitness overall. You're just changing the definition. It's that simple. And the current definition of fitness has led to innumerable predictions, the gold standard of science, that come to fruition. So, no, we're not going to change the definition of fitness. You got to try again. You got to come up with a different means by which to support genetic entropy, other than just going around and changing what things mean in science. Like, flat Earth will work if you change the definition of gravity. So, anyways, let's talk about Jeanson. And then we'll move on to the next chapter next time. So, Nathaniel Jeanson's got some problems. And the main folks that you want to talk to about that are going to be Dr. Dan, who's Creationist here on YouTube, and David Neff, who's also here on YouTube. You should sub to both of them. And they've done a fantastic video on why Nathaniel Jeanson is kind of effectively like a snake oil salesman, but also why he's just wrong. So Jeanson has in his mitochondrial mutation rate study, quote unquote, he's got this lovely paragraph right here. The only remaining caveat to the present results is whether the mutation rate reported in Ding et al. 2015 represents a germline mutation rate rather than a somatic mutation rate. To confirm germline transmission in the future, the DNA sequences from at least 3 successive generations must be sequenced to demonstrate that the variants were not artifacts of mutational accumulation in non-gonadal cells, so like autosome stuff. Now, this is from Jeanson's own paper. This is straight from the horse's mouth, so to speak. And what he's essentially saying is my entire work relies on Ding et al., the paper that I'm citing in my paper to get this mutation rate, mitochondrial mutation rate of 6,000 years, being germline mutation rate. So David Neff, one of the folks who's pictured here, thought to himself, well, why don't I just reach out to Ding et al. And ask them, was it germline? Were you guys measuring germline mutation rates? And so that's what he did. And I don't know why I didn't think of it myself. So here's what we get from Jun Ding. Thank you for your email and for the question regarding our 2015 PLOS genetics paper. We have 2 points of response. In our original PLOS genetics paper, PLOS, PLOS, I never know which one to say. We never tried to estimate mutation rates. It's in Dr. Jeanson's post that he used our trio to estimate mutation rate. This work was not done to determine rates of germline mutation, only prevalence of variants in the sample. In his blog post, he used 63, 35 plus 28 mutations from our data set to estimate mutation rates. We believe that these 63 homoplasmic mutants could represent a combination of germline and somatic mutations. So no, he wasn't using germline mutation rates. That's it. That's the ball game. Case closed, And I'm quoting Dan here on that, but we're gonna let Dan kind of harp for a moment on some additional problems here because I think he kind of pulls it together quite eloquently, honestly. So let's get the volume up so you guys can hear it.
That's the ball game. So I just wanna summarize this really fast, just to review what we've talked about. Jeanson is wrong about everything here. So he ignores selection, right? He ignores drift. He ignores all the other mutation filters I talked about, right? He ignores bottlenecks, right? And David described some of these. A founder event is kind of a category of a bottleneck. It's a similar kind of dynamic, right? He ignores that other parts of the genome have a larger effective population size, which means even if everything about the mitochondrial DNA is right, let's assume that everything that David and I have said so far is wrong. Let's assume that. Jeanson is still wrong because the other parts of the genome have a larger effective population size, which means the convergence date is further in the past. So if you put the mitochondrial convergence date at 6,000 years, the nuclear DNA is necessarily going to go back further than that, and that violates the Eck timeline. You can't squeeze all that stuff into 6,000 years. But let's put all that, I couldn't even fit all this on one slide. Let's put all of that stuff aside. And yeah, I see in the chat, rate heterogeneity. Yeah, let's put all of that stuff aside. Even on the one small thing, the you had one job version of this, right? Using the data from Ding 2015 to determine the mitochondrial germline mutation rate. Jeanson couldn't even do that correctly. For the reason that Jeanson himself identified in the paper where he claimed he could do that. It turns out that the thing that he said, by the way, this might invalidate everything I just said, it turns out that that thing is actually the case, and he couldn't be bothered to check ahead of time.
Exactly, and obviously he didn't bother to do his homework. I emailed Ding, and Ding was very kind to respond, and I really thank him for that. Why couldn't he do that? Why did he have to say, maybe it's this, maybe it's that? I'm just going to go with this because it helps my case.
Yeah. So
if you're going to wager a bet, I have a prediction, and my prediction is that he deliberately left out the African genetics and the Neanderthals, and he deliberately used this study, or misused the study, because he knows that if he included that, then his own analysis would be refuted.
Yeah, we didn't even talk about Neanderthals and how that blasts the rate too, because now the Neanderthal convergence date has to go further back. It's just a big giant mess. So the takeaway from all of this is that Jeanson's own data, the only data that he uses in this groundbreaking, paradigm-shifting study, right? His own data refute the conclusion that he drew using those data. Exactly. That mitochondrial Eve existed 6,000 years ago.
Yeah.
And that's all she wrote, guys. As far as the kind of Team SFT response to this, there's very little mention of the actual problem, which is, they'll go on, Standing for Truth responded, he goes on a lot about why ignoring drift and selection and the concept of substitution rates and all of that stuff, why that's actually not none of that matters. It does. It proves Jeanson is incorrect on a lot of stuff, but none of that matters in comparison to the brute fact that Jeanson said, if A is true, then my study is valid. If A is false, my study is not valid. A being that Ding et al. Used germline mutation rates. And A was false, so by his own admission, his entire work, deep sixed. That's it. Game over. Case closed. So, today what we have kind of discussed, right? What we've kind of gone over is how pedigree rates deep six young earth creationism. Because as it turns out, when we look at the pedigree rates of all of the great apes, including humans, they validate old time scales and they match up with fossil divergence times. We've talked about how the Y chromosome, especially in sequence identity, shows that chimps and bonobos are the most divergent, have experienced the highest mutation rate, which is supposed to be this deleterious genetic degradation in effect, genetic entropy in effect rather, not humans, and that in sequence identity, humans and chimps are still the most similar. We've seen how ENCODE, by their own admission, shows us that the human genome is not 100% functional. It's not even 80% functional. It's probably around 12, but Max is at around 25. This is, again, by the ENCODE researchers' own admission, the work of Dan Grar and others, and knockout tests additionally show us that no, the genome of any organism is not 100% functional. So that's 3 so far. And lastly, we've talked about how the mitochondrial mutation rate is indeed not showing us a 65 to 10,000 year old mitochondrial Eve. Not by Parsons, not by Jensen, and certainly not by Ding et al., who were, as you just saw, kind of appalled by the work, the way that their work was being used in kind of a miscarriage of science. And then we also talked about how genetic entropy just isn't a thing, and how it does not fly, it doesn't work. Now, in my work, in the papers that we've gone over, we went over the Toba eruption and showed why that bottleneck isn't legitimate and isn't thought to be legitimate by anyone. And we also looked at some genetics for the great apes, which is my specialty. But if you want to know more about genetic entropy, why it's bogus, and you want to catch the rest of that full video, which is, it's all immaculate, you should subscribe to Creation Myths, and you should subscribe to David F. You should subscribe to both of them. And I think that's going to do it for today. I think that's going to do it as far as this chapter of Standing for Truth covering genetics, and, you know, bolstering around like the cock of the walk that genetics supports him. Like, it's gotta be the most self-aware wolves thing I've ever seen in my life. It's so bizarre to me that you can be presented with this data as standing has been and still come to the conclusion that he's coming to, which is why, as others have said, his channel is mostly preaching, it seems. To each their own, I suppose. So thank you so much for being here, my gentle and modern apes. We'll probably cover the next chapter soon, because it's covering fossils, which I enjoy much more morphology than kind of the genetic stuff. But do take care of yourselves, and I will see you next time.